Hello, everyone, and welcome to today's webinar, Improve Whole Exome Sequencing Library Prep from Low Input and Degraded FFPE Samples. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Covaris. To learn more, visit covaris.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speakers. Joining us today is Dr. Nripesh Prasad, VP Scientific and Technical Development at Discovery Life Sciences. Also joining us today is Dr. Eugenio Daviso, VP Solutions at Covaris. For a complete biography on our speakers, please click on the Presenter tab. Eugenio, Nripesh, thank you for joining us. You may now begin your presentation.
Thank you for the introduction and good morning, everyone. My name is Eugenio Daviso, Vice President of Solutions at Covaris. Today, in the dynamic landscape of our company's evolution into a solution-oriented entity, I will unveil our vision to empower labs worldwide. Our aim is to equip lab with the tools and capabilities needed for efficient biomolecule extraction and molecular processing. I am excited to share how Covaris, a name synonymous with excellence in DNA shearing for library prep, is revolutionizing the approach to sample preparation, pivotal for genomics lab. Historically, the precision and reliability of our instruments to shear DNA have been instrumental in ensuring the high-quality sample preparation necessary for accurate sequencing data and bioinformatics. What's truly exciting is how Covaris today is embracing the burgeoning field of multiomics. We are expanding our technology prowess beyond the DNA, encompassing RNA and proteins. This holistic approach is crucial as we delve deeper into understanding the biophysics of complex biological systems and helping our customers and collaborators to unravel the intricacies of either healthy or altered biological states, thus delivering a robust solution for our customers and collaborators. Let's consider the challenges faced when working with blood or FFPE samples. These are common yet complex to process, often hindering the reproducibility and reliability of results. We are addressing these challenges, optimizing sample preparation to maintain the integrity and usability of these precious samples. We are committed to delivering solutions that are tailored to meet the core needs of our customers, focusing on three foundational pillars: enhancing user experience, delivering excellence performance, and optimizing cost efficiency. This diagram illustrates the crux of our workflows from sample to detection, focusing on germline, somatic oncology, and proteomics workflows, pinpointing the area in yellow where we are building not just products, but solutions. The blue arrows represent the component of the workflows that go from sample to analytics, divided into a simplified view of generic lab processes. To this audience, I would like to focus on two novel Covaris solutions ready in late 2024 and early 2025. The TrueCollect is a remote whole blood sample collection device coupled with an extraction kit. It offers a solution for stabilizing 50 microliter of whole blood sample collected for a finger prick and streamlines the entire sample handling process, from remote collection to DNA extraction and fragmentation. Its integration into library preparation for germline testing and studies is effortless. Covaris DNA library preparation for whole exome sequencing provides versatility, and it can be utilized alongside with TrueCollect with DNA sourced from blood, or with FFPE Smart Solution with DNA extracted from FFPE, or other extraction method you have in your lab, offering a single protocol for library prep that remains independent on the sample origin. The performance of this solution is consistently strong, offering ease of integration and cost effectiveness. We are actively seeking collaborators interested in enhancing their workflows through ours, and I encourage all viewers to engage with us. Your feedback is invaluable as we strive to understand your challenges and transform them into opportunities for improvement. As we move towards a more solution-oriented approach, let's take a moment to look at the full spectrum of our capabilities. On the left, we have outlined the fundamental sample types that underpin our technological advancement. For blood and FFPE tissues, we offer a suite of extraction solutions that greatly facilitate the retrieval of nucleic acid and of proteins. We offer nucleic acid fragmentation instruments and consumable, and soon a library preparation kit for all DNA sources. Based on the lab needs, we can tailor automation workflows that are easy and fast to integrate within a pipeline. Now, on the right column, where we see the broad range of applications that benefit from our technology. The application we service are currently integrated in academic, reference, clinical, and biopharma labs. Each one of these sectors has a unique demand and high standard that we meet with precision. This framework is our commitment to advancing science across all fronts, catalyzing discoveries and supporting our customers to develop life-changing therapies and diagnostics. Demonstrating our dedication to empowering our customers and advancing scientific progress together, we have partnered with LabRoot and our scientific collaborators to shed light on the remarkable discoveries and their profound effect on people. I invite you to listen to this recorded session to corroborate the claims made here today. In the first webinar, Jeff Conroy presented clinical data from the INSIGHT panels and its relevance to inform treatment decisions, including targeted and immunotherapy options, eligibility for clinical trials, and mechanism of therapy resistance based on the genomic and immunoprofiling of patient tumors. By working together with OmniSeq, we have made significant advancement to the Covaris automated FFPE extraction. Our collaborative milestones have been recognized through the exceptional performance benchmark set by the TrueSight Oncology 500 metrics as published in the PLOS ONE article. During the second installment of the webinar series, Dr. Serra showcased data provided by Quest. This data compared mechanical and enzymatic fragmentation methods across a broad sample set for all exome sequencing. The use of Covaris has proven to yield consistent results regardless of the diversity in DNA origin of the samples. Our methodology stands out for its ability to reliably produce precise-sized DNA fragments, which greatly simplify the library prep process by minimizing failures due to poor control of the DNA fragmentation. Such precision has far-reaching benefit, enhancing not only the performance, but also the cost-effectiveness of all exome sequencing workflows. During the third episode of our webinar series, Dr. Levine from MIT presented ultrascriptome data comparing the use of Covaris and chemical fragmentation of both intact and degraded RNA aliquots. The sashimi plot featured provide a quantitative visualization of RNA sequencing alignment derived from each method. This comparative analysis underscores the superiority of Covaris-based shearing. Notably, it reveals a marked increase in expression within crucial regions of gene clusters and untranslated regions or UTRs. What's compelling is that Covaris shearing not only enhances performance by ensuring data integrity, even in degraded sample FFPE, but also streamlines the workflow. As we continue to delve deeper into the intricacies of genomics and the impact it holds on the future of medicine, it is my pleasure to introduce Dr. Nripesh Prasad of Discovery Life Science. Dr. Prasad is at the forefront of biomedical innovation, where his work at discovering life science harnesses the power of precision medicine to tackle some of the most challenging conditions facing cancer and beyond. With an illustrious background that mirrors the commitment of discovery life science to provide the highest quality biospecimen and analytical services, Dr. Prasad's role is pivotal in propelling research and clinical application. We also take this opportunity to acknowledge the company's impressive growth, highlighted by this recent expansion at the Hudson Alpha Institute of Biotechnology, an achievement that truly reflects its deserved success. Please join me in welcoming Dr. Nripesh Prasad, a scientist whose dedication to advancing genomic and proteomics understanding is helping to pave the way for breakthroughs that will reshape our approach to health and disease. His insights today are the catalyst for the transformative research that lies ahead. Dr. Prasad, we are looking forward to hearing your perspective on how discovery life science is shaping the landscape of bioscience research in somatic and germline genomics applications.
Thank you, Eugenio, welcome. Pleasure to be here in front of everybody for this presentation. Today, I'm going to present a little bit about how we can utilize the Covaris AFA technology in whole exome library prep from low input and degraded FFPE samples. Before we jump into that, I want to just give a little bit, information on our company highlight. We are a prominent global brand since 2018 and headquartered in Huntsville, Alabama, within the Hudson Alpha Institute for Biotechnology campus. We are one of the leading commercial biobank and provide clinical capabilities with leading technologies across molecular pathology, genomics, proteomics, and clinical flow services. We have an extensive client base and trusted by top 25 biopharma and biotech firms, and we're deeply rooted in science and provide senior level scientist and pathologist support for all major projects. As you can see over here, we just recently moved into our new building, which is located in the campus of Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. This is a state-of-the-art building which houses the genomics, proteomics, cell biology, clinical flow, biospecimen, molecular pathology, cell and gene services, along with the donor clinic in a 93,000 square foot building. Our portfolio at a glance is separated into two major sections. One is the products, and another one is the services side. On the product side, we have the biospecimens, cell and gene therapy, and in vitro preclinical. The services side, it's mainly divided into genomics, proteomics, molecular, and clinical flow cytometry services. Jumping a little bit into the analytical validated assay into genomics, I'm just going to slowly move towards providing a brief background of the company highlight now into transitioning into what we do in genomics and how we do things in genomics. These are some of the analytical validated assays that are available for services. Includes whole genome sequencing, whole exome sequencing, using both the XGen IDD exome v2 as well as the Twist exome v2, poly-A library prep along with the globin mRNA depletion, ribosomal RNA sequencing, TrueSeq RNA exome, RNA fragmentation, low input RNA sequencing using the Takara Pico v2 kit, the TrueSight Oncology 500, cell-free DNA sequencing. Apart from that, DNA extraction from the whole blood using EDTA samples or PAXgene, dual isolation of DNA RNA from FFPE and biopsy samples, whole blood stability study, as well as the RNA isolation for the nasal brushing samples. Automation is at the center of our process. We see automation as a very deep and very reliable and, of course, very proven technology for what we do in genomics. As far as general processes, any tube-to-tube transfers, tube-to-plate, sample normalization, aliquoting, DNA shearing, bead purification, kappa qPCR, library normalization, library pooling, DNA extractions or from EDTA or PAXgene, DNA RNA extractions from FFPE or saliva samples are all automated across multitude of platforms. Some of the library preps have been automated, as you can see on your right-hand side. Includes the whole exome library prep, using both the IDT exome as well as the twist exome V2 whole genome, TSO 500 free DNA, TrueSeq RNA exome, microRNA library prep, the QIAGEN multimodal panel, the TSO 500 etched high throughput. This is from the solid tumor, and the Kappa Hyper Prep RNA library prep. We have a fleet of robots that includes, Beckmans, Hamiltons, and Cyclone robots. Jumping into the FFPE samples into the clinical research and focusing more on the topic of today, why we are here today, I want to take a step back and just lay out some of the brief points here. As we can see here is formalin fixed paraffin emitted or FFPE method has been utilized to preserve clinical samples for years at low cost. We are talking 15, 20 years, maybe more, for some of these samples. FFPE samples globally account for the largest archived samples. By some estimate, it tops around 1 billion samples that have been archived. That's a huge amount of samples. Apart from that, most of these FFPE samples are well annotated, with the patient clinical data or the histopathological data. These samples thus provide a great resource for the researchers. One of the drawbacks with the FFPE preservation that has been well discussed in publications and among scientific community is that it's like the FFPE preservation process leads to degradation of the nucleic acid. It's the cross-linking and the decross-linking, processes that are attributed to those kinds of degradation, thus making it challenging to generate a consistent and reliable data from these FFPE samples. As we move forward, what we are seeing is a little bit shift in, the clinical trials as more invasive methodologies are being let go, such as resection, and less invasive core-needle biopsies are a preferred sample collection method. What this creates is a need of a robust library prep protocol to not only utilize the degraded FFPE samples, but also, sub 50 nanograms input amount for both DNA and RNA sequencing at a constant basis. These core-needle biopsy samples are not that big, and thus provides both challenges as well as opportunities for us to produce meaningful data from these highly precious FFPE samples from clinical trials. I want to show this slide here to set the stage at which we start differentiating. The paradigm that DNA or RNA is gets highly degraded in an FFPE samples is a little bit of a to be challenged here. What we are showing here on this slide is our optimization protocol. As you can look in the figure on your left-hand side, there are two panels on that. One is a standard protocol, and another one is an optimized protocol. Clearly, you can see over there is that on the optimized protocol, 100% of the samples have a DV200 of 20% or higher. This is the RNA quality metric from an FFPE sample. Same FFPE samples using the standard protocol as well as the optimized protocol. It's a huge difference in that. So I would like to challenge the general consensus or the broad consensus among the scientific community as to, is the RNA really degraded in the FFPE block, or is the way that we are extracting it makes that difference? With this optimized protocol, I would like to mention here that We use the Covaris AFA technology to break the FFPE matrix, followed by downstream bead-based DNA and RNA extraction method from the same FFPE lysate. This protocol has greatly enhanced our capabilities, provide consistent quality data across multiple oncology indications, as you can see on the figure on your right-hand side, without any loss of RNA quality or DNA quality or quantity, by moving to a standard column-based protocol to a more robust, acoustic technology that allows us to get more longer insert RNA from these archived FFPE samples. I just want to jump into a little bit as to looking into where the clinical trial samples, majority of that comes down to whether broadly classifying into resection or core needle biopsies over here. Within that panel, you can see that we are consistently producing very ideal yields of nanograms per millimeter square from either resections or core needle biopsy samples. This method has been analytically validated in our labs and utilizes the Covaris AFA technology to lyse the FFPE samples. As you can see here, the average RNA yields are always greater than the DNA yields of the same tissue types. And it's a very known fact that RNA is a dynamic molecule, and thus the content is higher. So, we are expected to get higher RNA yields from the DNA yields. Different tissue types have different average DNA and RNA yields, as noted. This should be appreciated by the cellularity or the density of cells per millimeter square within these organs or tumor types. Actually, endometrial and ovarian being very highly cellular in nature. The amount of the DNA and RNA that is yielded from both these conditions are quite high as compared to lung cancer or any other cancer or gastric cancer types. What we are showing here is that if you look across both the resections and the core needle biopsies, we have a robust DNA and RNA yields captured across both collection methodologies. The key point of this is to make sure that we are measuring the amount of the DNA and RNA yield as to the surface area of the sample that went into the extraction. Let's take a little bit, to the DNA fragmentation approach. So what we showed here is that yes, Discovery has utilized the Covaris technology to generate a very robust DNA and RNA extraction protocols across multiple indications and both either resections or core needle biopsy sample types. The next step of that process I would like to discuss is a little bit on the DNA fragmentation approach. The DNA fragmentation is the most critical step of any DNA library prep method. Over the years, several methods have been tried, and broadly these methods are classified into either mechanical or enzymatic categories. One of the most prevalent mechanical shearing techniques is the acoustic shearing done by Covaris, where these sound waves are used to induce cavitation in samples, and it mechanically fragments the DNA. Enzymatic fragmentation uses different enzymes, depends upon which vendor you're talking about, and they are going to cleave the DNA at different points depending upon what the chosen chemistry of that enzyme is. These methods both have some benefits and I would rather not say drawbacks, but some things to consider for the downstream workflow optimization. I want to show you this this table here where some of the pros and cons are listed between mechanical and enzymatic shearing, and I'm gonna take my attention towards the the focused acoustic shearing technology from Covaris and compare it with a general enzymatic fragmentation side of the things. Covaris methodology is considered to be a gold standard in the industry for genomic DNA fragmentation. You have a perfect control on the fragment size, and the same protocol can be used regardless of the input material, input amount, or the complexity of the genomic DNA. That includes the GC content. However, on the other side, enzymatic fragmentation does provide a less upfront expense. To some extent, it is scalable. We can come to that. And it provides flexible reaction volumes, easy setup, things like that. But what we have to consider is what we are getting out of it, with both the technologies. The Covaris shearing technology does involve an upfront instrument cost, and I would like to talk about some of the scalability and sample tracking, side of the things here. However, on the enzymatic fragmentation, some of the processes, or some of the drawbacks are beyond cost. Actually, they're prone to sequence bias. They're sensitive to contaminants in the DNA samples, and they require significant optimization based on the input material, input amount, complexity of the genomic DNA, and things like that. As far as scalability and sample transfer and tracking goes, I think Covaris has done a very great job by addressing it with a new R230 instrument, which can be directly integrated into a liquid handler like an i5 and i7. The TPX plates improved liquid handling and can be used throughout the first bead cleanup. So you don't necessarily have to transfer your samples. You can normalize samples in the same plate, take it into shearing, take it out, and straight away goes into end repair, A-tailing, and ligation. There's an RFID plate tracking technology, being introduced, and overall, with these optimization and things like that, I would really like to challenge about the time and the scalability of the things. I think where we are right now with Covaris is with the introduction of the automation, even before the R230 instrument, our lab deploys the LE, and we were deploying the LE220 RSC instrument, and we had a way to automate the entire process of sample normalization to transfer into the Covaris plates of the 96 Covaris plates, the glass plate, and then transfer from the glass plate back to the PCR plates, and then continue into end repair, A-tailing, and ligation. So it's 100% hands-free liquid transfer methodology that we have that we have developed in our lab. The R230 does provide, and the TPX plate provide that kind of flexibility for people who would like to get it off the shelf now. And of course, it provides end-to-end sample tracking and audit proof, and as I said, samples are transferred to and from the 96-well Covaris plate using our methodology. So, having mentioned the utility of the acoustic technology over the enzymatic technology as a backbone, and where we have incorporated these acoustic technology into our processing, is very clear. One is during the isolation and extraction phase. Another one is at the genomic DNA, fragmentation or shearing stage. Now I'm going to walk you through a small experiment that we did and show some of the data between the Covaris and enzymatic shear testing that we have done internally, and some of that is quite obvious. Apart from all the advantages of automation to sample tracking and all those things, we really wanted to get down to the nuts and bolts of things and see, hey, what does these two technology when you put them head to head on the same sample type across some of these challenging samples. So as you can see over here on this in this page, is like it's a little bit descriptive. I'm sorry, the figures are a little tiny, but again, the key of the point is that it's like we picked up a couple of FFPE samples and worked across various input types from 200 nanograms to 100 nanograms, 50 nanograms, and 10 nanograms in triplicate, along with the NEB platinum genome samples. Deployed different GDNA shearing technologies. Mechanical shearing was done using the Covaris LE220 RSC instrument. In the enzymatic testing, we went with vendor N. The downstream library prep was done with the NEB FS2 kit for the library prep, followed by IDTV2 exome kit for hybridization. As you can see over there in the center of the figure is like the Beckman I5 automation. This entire process of the library prep, exome capture, wash, and the final amplification is 100% automated on our Beckman instruments. Both I5s and I7s have the capabilities to perform this assay for us for both either kappa or IDT exome capture, and even the twist exome V2 capture. The sequence is on the NOVA 6000. We're targeting approximately 150 to 300x coverage with the IDT-V2 exome baits. And we basically were looking into very basic key metrics here. What was the Q30? What was the insert size? What was the discordant percent? Because the downstream application of that data has to go through these what you call as the sequence QC metrics. So just to show over here, the sequencing QC quality of that is that comes out in Q30. We all know what Q30 is. I mean, it's parsing needles here between the Covaris shearing and the enzymatic shearing, and what we can clearly see is that it's like across FFPE or even with the NA128 samples. The Q30 bases were very comparable between the Covaris and enzymatically sheared samples. Not a big difference in that. I would like to point towards this this slide a little bit. Might not seem significant difference here, but it is very consistent across the board. And what we see here is that the library insert size, was definitely higher across the board, across different FFPE samples, all the input, including the NA12878, for the Covaris shearing as compared to the enzymatic shearing. Now, this is going to be very critical for the downstream, metrics evaluations of coverage and all the other things. But what we can clearly see over here is even especially if you look into the NA12878 samples, which is the platinum genome samples, there is a much more tighter control over shearing, using the Covaris, where we can consistently target for a bigger insert size as compared to the enzymatic shearing samples. This again comes down to the mean target coverage, and what we see over here is that it's either for the enzymatic or the mechanical shearing, after all the dust is settled, and now we cover for that. This may not be that much significant because ultimately the mean target coverage also depends upon how deep you sequence, things like that. And between those metrics, I think this can be easily controlled by doing top-ups if necessary. This is something that I want to focus on actually. Very clear here. The discordant rate. This was very clear that apart from the Q30 being very close to each other, the sequencing quality being close to each other, the insert size moves a little bit in favor of the mechanical shearing than the enzymatic shearing. The coverage, kind of the dynamics of how deep you want to sequence it. The discordant rate is something that is going to be inherent to the points towards the technology that we are using here. It is this thing is very clear to us that the Covaris shearing samples consistently kept the discordant rate very, very low as compared to the as compared to the enzymatic shearing that we're using here. What we're seeing here is like, even with the NA12878 samples on the far right-hand side of this of this plot, the enzymatic shearing it runs higher in discordant rate as compared to the Covaris shearing samples. The Covaris shearing samples is very consistent for the platinum genome, and that is something that is something to be well aware of, that is going to show up in the data, and hence some of these choices have to be made as to where we want to go with the with the methodology that we are going to choose. We looked into some of the accuracy and precision for the NA12878 samples across multiple input types for both enzymatic and Covaris shearing samples, and the precision and accuracy for the NA12878 samples are very, very spot on for either enzymatic or Covaris shearing samples. Not a big difference there. However, if we have to summarize between all the things that we see here, the library insert sizes are greater with the Covaris AFS shearing compared to the enzymatic shearing samples. We have seen this across the board, across multiple sample types, across multiple projects, and across multiple testing internally, and that has been a very consistent feature for that. We do not have to optimize or tweak our protocol or change in our methodology for different source of DNA or different extracted material or elution buffer. Covaris methodology is very much adaptable to any of those variables. The Q30 base is comparable between Covaris and enzymatic shear samples, and we did see high accuracy and precision between for the platinum genome. However, I would like to point out that the discordant percent was quite higher across all input types for both FFPE and NA128 sample processed using enzymatic shearing as compared to the Covaris AFS shearing methodology. This should be looked in very carefully. That higher discordant percent of 5% to somewhere around 10% at times will have a very, and we all understand that is going to have a very dramatic effect on the accuracy of the variant calling and things like that, especially from the clinical trial FFPE samples, where they're highly fragmented, highly degraded, and they have to be evaluated based on that. The high discordant percent points to the highly biased call from FFPE samples, and thus technical artifacts affecting the robust genotyping calls with enzymatic shearing is going to be part of the process. What I would like to say is that it's like taken all together, our implementation of the Covaris AFS shearing in isolation and library prep from degraded and limited input material provides the best meaningful data across the board. I would like to acknowledge a couple of folks from our lab. This is not the entire list, but I would like to just call out Rachel and Rebecca. They lead our DNA and isolation queues respectively, and they have been a key member of our team to help work on this project and testing. And we're with the help of Melanie and in the automation, and Boris and Michael for the downstream bioinformatics analysis, I think we have settled on our plan for the incorporation of the Covaris technology across the board from both isolation and the library prep. I would like to thank you for all to attending this webinar today, and open to answer any questions you have. Thank you all.
All right. Well, thank you so much, Nripesh and Eugenio for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here. So this first question I have for you, Nripesh, how did you determine the discordant rate percentage? And did this calculation include various technical replicates and concentrations?
Yeah, thank you. I think if I can you guys see this slide?
Yes.
Okay, thank you. So I just wanted to put this out there. Actually, I was putting some data together, and this thing is like, I think it's going to explain a lot to a lot of people actually right up front. And that is, as you can see over here, these are various FFPE samples. I think there was like 4 FFPE samples in triplicate across different input types, and these are across 200 nanograms, 100 nanograms, 50 nanograms, and 10 nanograms input. And utilizing the either the enzymatic shearing or the Covaris shearing for the methodology, and the pipeline we just showed exactly same IDT capture, IDTV2 capture for whole exome sequencing. And as you can see here, as you go down on the input amount from 200 to 10 nanograms of FP samples, the discordant rate keeps creeping up on you using the enzymatic, as far as the mechanical shearing with the Covaris AFA technology. It stays very stable actually. Very, 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 very consistent, and across the board. I think that is where the return on the investment, which I think the market argues that yes, it's cheaper, but my question becomes is it better? In our incorporations and across multiple projects and multitude of samples we have processed, we have very much settled on the Covaris as a part of our genomic DNA sharing app. And then to take that answer to one more step is we have utilized Covaris actually not only in the gDNA sharing, it's very early on into our processing. From the extraction per se, our success rate of sample processing from starting from sample, and especially these precious clinical trial core needle biopsy samples, which often also require macro dissection to generate a meaningful data which has very little noise or background noise that can be that can give the confidence of whether or not you should be trusting, or how much you can how much bias is there in the data set. I think coupled with both the steps of implementation, I think it's a very, very good model for us.
Great, thank you, Nripesh. This next question asks, what about skin tissue samples conserved in FFPE?
Skin tissues, my favorite. Yeah, I think I don't know if the data showed the skin tissues or not, but we have processed quite a good amount of skin tissue samples. We are one of the partners with one of the with the Aster Insight program, which used to be previously known as Orion Avatar program, that is a network of, I think, 14 or 16 cancer centers that pool samples, and we extract them and we sequence them, and skin is very much part of that program. And I don't think we have seen any trouble getting into or any difference of modifications of the extraction. I think my point is that it's if your extraction is controlled and you can demonstrate that we can extract good amount and quality samples from the FFPE samples, I think a lot of the failures happen in the extraction steps. Either the industry is not able to show that they have a robust extraction method of the FFPE, a quality extraction method, and a consistent, automated method for extraction, which we have demonstrated over and over. I think everything starts at that point. Once we get into material available, I think much of that has to be done at the extraction phase. Our lab has done a fantastic job in adopting Covaris. I mean, we adopted Covaris into our extraction back in 2016, I believe. Very much at the forefront of jumping on that technology, because we really saw the benefit of that technology. And as I showed from the data, one of the slides was that it's like showing off, just touching on the RNA quality. I mean, absolutely a game changer. I would say that for our purposes, because I mean, we have 100% success in that experiment. I mean, our success rate right now for FFP extraction is upwards of 95-96% of the samples that hit our doors ends up generating data, and then meeting the metrics or end deliverables, whether it's coverage or GB data, whatever the output might be.
Great, thank you. Now, can the R230 be automated on the Janus G3 liquid handling system?
Yes, I'm gonna answer to that. So we do provide automation services for integration on our workflow, and as a matter of fact, we have a dedicated team. It is possible to integrate it on the Janus under a modular approach, but this is the kind of question that requires a more in-depth evaluation, and I would encourage to reach us out so that we can have a discussion and really understand what the needs are for the integration, from number of touch points, from the different capability of the liquid handling system to be integrated with the R230.
Thank you, Eugenio. Another question here. Do you run the exome prep with Covaris TPX plate? And if yes, which volume do you use? Excuse me.
Oh, well, the Covaris TPX plate. Our protocol, we have been using Covaris for our shearing method since 2010, I believe, or maybe before that, actually. So it's a very old relationship with Covaris that we have been, hanging on to that. There have been recent iterations of enzymatic shearing that comes across, but anyways, not touching on that again. My point is that it's like we have introduced automation well before. I think a lot of people out there were putting in automation, and our protocols runs on those. I forgot the name of the plate, but it's a glass plate that holds a good amount of volume. So we sonicate, I believe, in 50 microliters on those glass plates, 50 or 52 microliters on the glass plates, with a couple of microliters for the transferred, dead volume and things like that. But we have been using that. But our lab has used the TPX plate. I think I have to check into what volume goes into the TPX plate for that. But we have used TPX plate as well. But I think for a routine purposes, the majority of our processing goes through straight up, with the traditional, the glass, the glass 96 plate of the Covaris plates.
Great, thank you. We're getting so many great questions in from our audience here, and we do have some more time, so we'll continue. This next question asks, in your experience, what is the primary advantage of incorporating Covaris into your workflow? Is it the reduction of failure rates, the flexibility of the process, or the ease of integration?
It's all of the above, actually. To be very honest with you, as I said, I mean, there are two parts of the process. One is extraction, and one is library prep. I think you cannot discount one for another. I see one of the questions also on the follow up on the skin is are you extracting with Covaris and then using? The answer is yes. Let me put it this way: our entire FFPE extraction, whether it's a single extraction, means either DNA or RNA, or dual extraction, deploys Covaris. We do not use any traditional column-based. I'll say this on this call, we don't use all prep. I think the standard protocol is pretty much the all prep, very much widely used and very much widely in the industry out there. We have found that the plot that I was showing was the standard prep, and and the optimized prep is all prep versus using the Covaris technology. And it's we found that back in 2016. even before, I think, Covaris has a very commercial product out there to utilize that. So I think, we do see that it is a less failure rate, ease of utilization, integration with automation. And I understand that that might not be possible for smaller labs, but I would highly encourage that to explore those opportunities within your university systems, core labs, or talk to us, or some people who can provide that opportunity, because ultimately it's about, either the clinical data is very important. I would rather not go with any like an isolation method. If I look back since 2016, we probably have extracted more than or close to easily 500,000 to 750,000, close to 750,000 FFPE samples easily, no doubt about that. And then consistently producing very high quality data, irrespective of the collection or sample or tissue type, whether it's skin or hard to digest or things like that. There's a little bit modifications that you have to do depending upon the tissue. Of course, skin being one of them. Hard to digest. So not going to go into the secret sauce here, but you have to play a little bit with some of the sonication steps there. But outside of that, it's very much feasible.
Great, thank you. All right, so you are extracting with the Covaris from FFPE skin biopsies and afterwards also shearing with the Covaris. Or what extraction do you use prior to Covaris?
I think I blended question number, two, it's like last question and thinking the same one. The answer is yes, we use Covaris at both the steps of extraction as well as gDNA shearing for every single FFPE sample that we process or, through the lab.
Great, thank you. All right. Now, what tissue pathology, tumor or non-tumor, was this DNA protocol tested on?
This was tumors. These were, I think I can, if you can contact me, I can provide you with some of the context of which tumor tissues or which FFPE tumor types were processed here, but these were tumors.
Great, thank you. Next question here. How is the ALU score calculated, and how can this score help compare results from enzymatic shearing and mechanical shearing in FFPE samples?
So ALU is kind of a known assay in the field out there. It's pretty much used there's an off-the-shelf assay that there was a company called Swift, and now it was bought by IDT later on. So they have this assay which is a qPCR based assay. So basically it deploys a short primer and a long primer to find out what is your how much fragmented, and it provides a very great calculation of the range is from 0 to 1, just like a RIM score or a DIN score. Now you can also utilize if you don't have access to that and you don't want to go too deep into the qPCR based or basing of the quality of the DNA that is coming out of the FFPE samples or any of the sample type, you can deploy just tape station. Tape stations are good instruments, workhorses. I mean, they're not that fast, but I mean, they're good. We do deploy a tape station as well on all the FFPE or all the genomic DNA that we extract through our lab. That's a very good DIN score that come out of it. I think you usually, we use either or both of these scores to evaluate the amount of degradation that is there. Now, I should say that the highly fragmented DNA, that means that the very low on the ALU score or very low on the DIN score, doesn't mean that Discovery doesn't have a protocol to manage that. It would just has to be tightly controlled on the insert size expectation that is going to come out of that, because I mean it's just fragmented. So we have to a little bit control on the bead purifications that you deploy during the cleanup processes in the libraries to just make sure that you can let go. But it's a compromise between diversity of samples versus insert size. So I think some of that I think has to be looked carefully into where the return on investment, or think how much you want to trade off between those two aspects.
Great, thank you, Nripesh. All right, we've got time for a few more questions, so we'll wrap up after a few more here. This next one asks, can you please share some bioinformatic observations which highlights that AFA provide more information for whole exome sequencing or genotyping analysis?
Yeah, sure, absolutely. I think I'll take that question as a rain check. I don't have the data here, but absolutely, I think if people contact me, I can definitely we can discuss it. No problem in that.
Great, thank you. Now, Eugenio, I have a question for you. Has Covaris developed or plans on developing a nuclei extraction approach from FFPE samples for epigenomic profiling purposes?
Yes, we do have solution for nuclei isolation from cross-link material, also from FFPE. And as a matter of fact, we have also a kit that enables the shearing and fragmentation of DNA from FFPE. Once again, being a solution company means to talk to us so we can really understand what the actual need of the workflow and the outcome of the analysis that is requested is crucial. I would really encourage this viewer to talk to us so we can show all the material that we have put together with the studies that we have done through the years to see if there is a common ground that we can help.
Great, thank you. All right, we've got time for one more question here, so we'll wrap up after this one. Which kit do you use for FFPE NA acid extraction? And is it the true extracts or column magnetic beads?
We favor magnetic beads over anything else in the industry. The kind of the operations that we run, we have a very heavy footprint in sequencing, and it's not possible for us to have column available for going in there. It's very hard to automate those things for us. I think as I mentioned, automation is the key to that and center of that. We have protocols and methodologies automated even before they have been realized out there. I think magnetic beads is as a principle of choice is key to us. And our kind of that blends very well with our interest and our expertise in automation. But I mean, we have been always with the magnetic bead sites of the true, the what's the name of the kit, the true pack assay kit. Yeah.
Great. All right, well, that will conclude our session for today. So Eugenio, Nripesh, do you have any final comments for our audience?
I would like to thank Nripesh for being with us today. And for the audience, I would like to thank everybody for calling in and listening to us. We look forward to talk to you, to collaborate with you so that we can keep improving our solution space and the solution that we can deliver to help each other to grow the genomic science even more. Thank you.
And I would like to add to that what Eugenio said. I would like to thank Covaris for all those years of collaboration that we have had. I think it's a very fantastic not only a technology but also a fantastic group of people. And people who know me know that I'm not generally a big fan of those things don't come out easily from me. But again, I think this thing has this technology has got its its benefits. If you go past the cost scenario of that, I think it is going to yield wonderful results. Beyond imagination from other traditional or what's available out in the market out there. But again, thank you all. I see there were other questions out there. I'm sorry we couldn't get to it, but more than happy to get into detail. Thank you for this opportunity, Eugenio and Covaris, and of course, Kaylee for the opportunity. Really nice. I really enjoyed this discussion, and looking forward for many more. I will be at AACR. Just wanted to put that out there. If you guys want to connect, please shoot me an email or throw me a and some kind of message on my LinkedIn, and let's connect at AACR.
Great.
Thank you.
Well, thank you again, Eugenio and Nripesh, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Covaris, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.